Hi,
Welcome.
everyone. Yeah. Hi, Jane. Thank you very much, Jane, for such a kind introduction. And so I'm going to give you an overview of the two packages we developed called CRISPRseek and GUIDEseq for design and evaluation of gRNAs in CRISPR-Cas9 genome editing systems. And as most of you already know that in this year, the Nobel Prize in chemistry was awarded to Emmanuel Charpentier and Jennifer Doudna for discovery and the CRISPR system for editing genome. In 2012, that's in 2012, they discovered that, and this year they were awarded the Nobel Prize. So what is a CRISPR system? CRISPR system actually is discovered in 1989, and in E. coli as in the beginning, it's just as a repeated, interspaced repeats, and subsequently described in different bacteria. Only in early 2000s, and it was discovered as an adaptive immune defense system found in prokaryotes that protect them from the macrophage and virus. And basically, and when the first time the virus invades the bacteria, and part of it is a sequence called the protospacer, and got introduced or incorporated into bacterial chromosome called CRISPR. CRISPR is for storing past invaders sequence called protospacer, and interspaced by repeats. And when the same type of virus invades again, and this CRISPR system then can recognize the protospacer and thereby cleave it and then destroy the virus. And in 2012, as I told you, that Emmanuel Charpentier and Jennifer Doudna, and as well as George Church and Van Zang's lab and several research groups discovered that this system can be engineered, not just targeted virus, but it can be engineered to cleave and even introduce new sequences into various genomes, including eukaryotes. So I want to give you a brief overview what's this system about. This system, for this figure, it depicts two major components of this CRISPR system. This is in engineered form for the most widely used and well characterized CRISPR system from the SP and S. pyogenes, which is a type of bacteria, and it's called CRISPR-Cas9 system. And so the one of the components is a protein, Cas9, which is a nucleus. As a nucleus can carry DNA, so it can it can cleave the target sites. And in addition, this component can recognize a short sequence called a PAM, which is immediately downstream or upstream. Can be upstream, but for this system, for this typical one, as a downstream of the 20 nucleotide target sites. And for the other component, which is depicted as like string, is the sgRNA or gRNA or single guide RNA. And from now on, we're going to refer to as sgRNA or gRNA. And it also consists of two components. One is constant region, which is from the bacteria. It can form stem loop structure and as scaffolding. And the other part is depicted as green, and it's called target specific region, and we refer to as gRNA, which is the part we're going to design for different research purpose. And this part will be engineered to base pair with the target site you intend to make changes. And the target site is depicted here, as target sequence, which is 20 nucleotide long. In addition to that, there's a PAM sequence required downstream here for this SP-Cas9 system, it's NGG. So only when both sides of the target sequence recognized and bound by the CRISPR system, and then the Cas9 will make double-stranded break. So once the double-strand break is made, and the DNA repair pathway will be mobilized to make intended changes as like using homologous dependent repair pathway if you provide a template, or make a gene disruption by using the non-homologous end joining pathway, which is error prone. So that's why I can introduce indels. So in general, it's quite easy, right? What you would think it's easy to design gRNA because all you're looking for the target site is whether there's NGG and PAM sequence. However, when you introduce and the nucleus into the genome, the other consideration is you avoid it cut anyone elsewhere. We call it off targets. And you also want to maximize the cutting in frequencies in the target sites because not every different sites will have different cutting frequency. And so the overall goal is to increase, or maximize the on-target coverage. We call it efficacy or efficiency. And with lower off-target cleavage, you can say high specificity. And so what rules governing this efficacy? And Dr. Ruth's lab studied about 1,800 guide RNAs turning across all possible target sites for 9 genes. And they constructed a predictive model using 72 features, including GC content, mononucleotide variants, dinucleotide and dinucleotide variants. Here it's a figure just depicts how different single nucleotide variants affect the cutting efficiency or efficacy. And so they used the guide RNA and the PAN sequence, and also flank region for minus base pair and then +3 and downstream. And they found that this different content or different variants of single nucleotide can affect the efficacy significantly. And the top one is favored, and the bottom one is disfavored. As you can see, for example, as arrow point there, and the G is highly favored and C is disfavored in the position 20. And also in downstream of the PAN sequence, there's also preference. So this is the one. And then, so in terms of off-target cleavage effects, and Dr. Zhang's lab studied about 700 guide RNA variants for 15 target sites. And so they basically create a panel of guide RNA variants with one mismatch for different position. They found out that not only mismatch number, but the mismatch position also affect the off-target cleavage. And here is the heat map for a guide position. Maybe I should have shown cursor so you guys can see. For example, the green means there's no effects, but in the seed region there's a high effect as a red. And in addition, they also discovered that even though the preferred PAM or canonical PAM sequence NGG is preferred with the highest cleavage efficacy, and actually the other alternative one called NAG also has a reduced effects. So when you search for guide RNA or target site and you want have search for NGG in the canonical PAM, but when you search for off-target, you would want to consider NAG as well. And later on, Dr. Ruth's lab constructed an even bigger data set, about 4,000 guide RNAs with one mismatch. This time, not only they change mismatch number, but also like a mismatch, not only mismatch number position, but also mismatch type. For example, A to C, C to G, is that going to change much? They found out that actually mismatch of type also make a huge difference. For example, this position, there's, no matter where, it's not position dependent. So it doesn't make difference if you make this change, G to T. However, if you, like here, if you make a change, and it's going to be the change, it would be based on no cleavage anymore. And the other type of changes, they are position dependent. Different position, here's a huge effect, there's no effect. So they create another model to do this kind of prediction. And in addition, they not only confirm that NGG is the preferred one, and their NAG is have reduced effects. They find additional NGA or NGT has effects. So this is an updated model, and so this system has evolved very quickly to increase the specificity, also increase the targetable size. For example, if you only have an SP-Cas9 type of system, you only can target NGG. How about those regions that has T-rich, or they have don't have GG PAM, right? So you want to target those sites. So there are a lot of variant different species have been discovered, which will have different PAM preference, different PAM orientation even, or PAM lengths, and different guide ion lengths. And in addition, there's a paired guide ion have been developed that configuration so that it has a nickase instead for that make like a Cas9 and nucleus that can make a double strand break. Instead, it will nick the DNA and make one strand cut. So this kind of paired system that can increase the specificity because you require a longer match, right? So this system will require you to have a different guide design because they want to design paired, how their configuration, what's the distance between them, what's the orientation, different, there are different nucleus, focal 1 versus Cas9, and CPF1, which actually has a TT reach inside, so that will require different thing. Also, it has, instead of PAM, it's not in 3', it's in 5', and there are high fidelity Cas9s, and then truncated Cas9, which is low, small, less or shorter guides. So with all these changes, and in addition, recently there is more to be added to the toolbox. One of them is called why I cannot. Can you guys see the title? I cannot see the title. I don't know why. Can you guys see the title of the? I guess I cannot. Assume you guys can see. And this is a base editor. So there are two types of base editors that developed by David Liu's group. So why they're doing this? Because for Cas9 and SP-Cas9 system, it actually increase, as I mentioned to you, you make double strand break, which can introduce indels because of non-homologous end joints. It's actually error prone. So here they try to use the deaminase to attach to the system that will not make a double strand break. And this way, it will not introduce this error or indels. Instead, it will have made a precise edit from, for example, from CT to AG or AT to CG. And this will require different design because the window, you can think about within some certain window, you don't want to have the same base there. You want to avoid the off-target changes. Another recent one is called prime editing system, which leverages a reverse transcriptase and have a PEG RNA in addition to a standard gRNA as a guide RNA. So PEG RNA will have encoding not only the edited sequence, a targeted sequence, but also the binding region and as well as the intended changes. And all these changes make the design of the system more challenging than we initially thought, right? We would think just looking for the PAM. So we designed CRISPRseek with all this in mind to be versatile and adaptable and also flexible to meet the needs of whatever changing requirements. So for our system, we not only can define guide RNAs with high on-target and low off-target cleavage using the scoring methods or the algorithms developed using the experiment data. And also we can design guides for base editor and also prime editors. And in addition, we've been configurations such as novel configurations such as the paired nickase, Cas9 focal 1 dimers, and as well as different Cas9 species as CPF1, IM, and Cas9. And we also accommodate different methods for synthesis or delivery because the different system will require the constraints of different guide. For example, for U6 promoters, you would like to have the G as the first first base, and we can do that in this system. In addition, there's actually this is from one of our collaborators that they're interested in target one allele on the diseased allele, but let the healthy allele alone. So we designed the function basically to target one allele but not the other, and also can target both allele if it's required. So in a nutshell, and to make it easy to use, we wrapped all the functions into workflow function called off-target analysis. And basically, this function is the most often used. It's for guideline search. If you just want to find out, if your target sequence contains some guide-RNAs, or you want search guide-RNA as well as off-target search and scoring, and also annotation, and whether they are inside the exon, whether inside a gene of essential gene, and also where is the sequence. And another one is called compare to sequence. I show you that it's for identify guide-RNAs that specifically target one or both of the sequences. So here is a simple workflow. In the default setting, is for the most well characterized SP-Cas9 system, and all you needed to do is input your sequence file as FASTA format or as FASTQ file. And then if you want off-target search, you can just input your genome sequence as well. And if you want to annotate the target and off targets, then you input transcript DB or organo. And this is one of the advantages of developer CRISPRseek in bioconductor, because the transcript DB, organo, and BS genome updated twice a year, and it's also very easy to make if you want to have your own transcript DB or BS genome. There's tools to do that. As I mentioned to you, there are novel configurations such as paired configuration. For paired configuration, and this one like SP-Cas9 one, in case, and all you need to do is just say you want the paired equals true and set up. This is a default for a system, and their PAM should be outside, so automatic search for you. And there are another system, since time limit, I'm not going to show you, but you can find out this in different system. It's annoying because I cannot see the title. It's too bad. Okay, how do I find the guidelines? Okay, this is if you're working with a different system, because the default setting is for the mostly widely used one called SP-Cas9, as I told you. And if you are using a different Cas9 case system, for example, CPF1, then you will change all you needed to do, you only need to change the PAM size to 4 instead of 3, and then you change guide-RNA size, and also the PAM sequence and the location. And for some people working on Cas9, for example, the PAM size is much bigger, and all you need to do is basically change these parameters, and they will output 4 different files for guide-RNA. By default, the CRISPRseek use the score method for Zhang's lab for the cleavage score, and the rule set 1 from rule lab for the efficacy. And the reason using those the cleavage score with Zhang's lab is because many species don't have off-target prediction algorithm yet. It's not because the algorithm, it's just because lack of data, right? So once we have data, we can plug in. Right now, have that as default. And I recommend that people actually using the cleavage score called a CFT score from the second data set from rule lab, because it's have a big data set. So all you need to do if you want to do that, just score method equals safety score, and you can change the rule set. Recently, I got a request, people want to use a more updated one, so you can just change to, you know, CRISPR scan or rule set 2. So it's quite adaptable and easy to modify or change. If you have anything you are interested in, and we can add it, or you have your own method plug in, and you're welcome to do so as well. And for base editing, as I told you, and it's a recent development, it's last year, it's like you can just change, you know, change base editing to true, and you can change what you want, what's your target base. By default, this is a window, editing window, and it will search for, you know, within this window for the editing window. And also you want to avoid those off target here. It's automatically done for you, and all you need to do is to change base editing to true. And you can imagine in the future there are different ones, so it can be easily changed. And for prime editing, it's more complicated because they are PEG only. You need to design PEG only. Also, another one is guide only also, so they are paired too. So this, however, for our CRISPR system, you can only need to change prime editing to true, and all these parameters is set for you. And by default, using the experiment data, we think that's the best setting. But in the future, if more, you know, refinement have been made by researchers, and then you can change easily, or you want to compare your own. Maybe you have one experiment, you could do that. And all you need to do is say, you know, what's the length of the sequence I want to change? You want how many base you want to change? What's the base of the target end? You know, all this is very flexible, easy to use. So I'm just going to show you quickly, you know, what's the, let me see, what's the output look like. This is a summary output, and so basically summary output will find the GRNA and plus PAM in your input sequence, and where is the in the genome, and this is the guide, and what's the target efficacy. So what this is, you want to be high, you want a high efficacy, you know, this I highlighted red, there's some reason I'll tell you. And then this is the highest. If you're only interested in, you know, knock something out, you really don't, you know, you don't care about off target, you know, then you can pick the highest one, because some system you really just want to get your screening for the mutation. And you can look for top 10 or top n. You can change that top n to 100 or 5, whatever number. This is quite flexible, it's dynamic. And you want to minimize this off target cleavage score. And these are 2 recent features I just added. This is actually entropy and frame shifting. So for a frame shift, if you want a mutation, you want the frame shifting, the high number of frame shifting, the largest of 1. So you want to be the highest if you want to make a mutation, because some mutation may not make much changes, right? And the entropy is channel entropy for measuring the mutational outcome complexity. And this is very useful if you are working with a CRISPR system for the tracing, like molecular tracing, and which will be helpful. You want to generate a lot of different ones so that you can trace lineage. For the off-target, this is detailed off-target. You can see that there are, you know, off-target sequence, whether it's in intron, in exon, what's the end is a gene you want to see, and what's the, you know, score, and you want to, you know, less score, list score, and then this is basically the off-target score, and number of mismatch, you know, what's the mismatch to the distance to PAM and alignment, and whether it's a PAM sequence, it's a 1 means NGG, and what strand, and also, you know, the chromosomal location. And so when after you look at the summary, you pick one, and one guide, you want to look at the detailed off-target score to make sure there's no essential gene here got knocked out by, you know, accident. And also you want to look at whether those off-target sites, one of them has a huge score and it's in exon or something. So this is important, not only look at that. This is the detailed and predicted, this is the predicted, okay, detailed mutation outcome. Basically, what do you this is like based on the sequence feature around the cutting site, 30 base before and 30 base. This is based on Lindell algorithm, based on microhomology and also sequence features around the cutting site, which is for SPCas9 17, 18 position, and how this is the sequence without editing, what's the outcome look like, and what's the frequency and where's the location. So it's very important for you to look at this especially if you're interested in see some outcomes and then you look at a prediction, it didn't come out, right? So you want to avoid that. And the second workflow is very similar to first one, except that I compare two input sequence, find the one that's specifically targeted for one instead of the other, or target both. All the parameters that I talked about apply to here too. And you simply, however, I want to say that after this, you need to once you identify a guideline of your interest, is either target the specific one, one sequence or both. You want looking for genome also. You basically call the first off-target analysis workflow again, just not like to make sure there's no genome-wide off-targets, right? So this is just to make sure. And so since because of time limit, I want to quickly say that for clinical and therapeutic application, it's very important to evaluate the outcome and not just predict, right? So this is one of the widely used method called GUIDE-seq for measureless, and basically use the erroneous non-homologous end joining mechanism to co-introduce a double-strand, oligos to the target cleavage site, and then you measure it. And it's experimentally it's quite easy. And however, the analysis is not quite complicated. We created a system called GUIDE-seq package and can do GUIDE-seq just to make sure it's easy to use. We just have two workflow function, GUIDE-seq analysis, and mostly you're going to use that. Combine off targets just to compare different experiment to pick. And so here, if you just look at this paper, and if you need to have any questions, let me know. And to make it easy to use, we have pre-processed scripts for you so that it can this script will generate the file needed for this GUIDE-seq package. And to make it even easier to use, we also developed a Gsp process that can take the run folder directly and get an input for GUIDE-seq. And so there are references here, and if you want to take a screenshot of this. Okay. And with this, I would like to thank my collaborators, and they contributed significantly to this work. And I don't want to mention all of them, but I want to thank especially my collaborator, Mike Brodsky, and Scott Wolf, and also two graduate students for GS. They basically single-handed created the Gsp process, Henry and also Tom. And also want to thank my conductor and core member Hervey Pages. He helped to speed up the CRISPRseek and the GUIDE-seq, and also Genotech Mike Lawrence for the GUIDE-seq contribution, and also Zhang's lab for I really thank the opportunity to speak here, and thank you very much.